Hey guys, Mr. P. This video is all about carbohydrate structure and function. So the first thing we need to talk about is carbon from an atomic standpoint. Now, in past videos, we have talked about atomic structures, so we're not going to spend a lot of time talking about how the atom is physically structured. But if you remember, protons and neutrons are kept in any atom's nucleus, and the electrons are kept in the outer energy orbitals that kind of surround the nucleus of the particular atom. Carbon has an atomic number of 6, which means it has 6 protons. We will go ahead and add the 6 plus signs to these red structures. Those would indicate protons. Carbon also has 6 electrons. Electrons are negatively charged, so we'll add the negative symbols to the blue structures. And then it would have 6 additional neutrons in the nucleus, which gives us the atomic mass of 12. But when we talk about carbon from an atomic standpoint, the inner electron orbital is completely filled. All atoms want to have 2 electrons in that inner energy orbital, and so all 4 of the additional 6 would be in the second or the outer energy level. Atoms want to have a total of 8 electrons in their outer energy level, and so that would leave 4 binding sites that this particular atom would want to fill in order to make that outer energy level full. That is critical in why carbon specifically has the ability to make and has become the main component of all of the organic biological molecules. Methane, CH4, is an example of what can happen because of the 4 binding sites in this one particular atom. 4 of these electrons would have come from the carbon, and the other 4, this one, this one, this one, and this one, would have come from each of these 4 hydrogen atoms that they are sharing. So these would be 4 covalent bonds. Because the electrons are being shared, and so that creates a CH4 molecule. Now, carbon doesn't only make covalent bonds with hydrogen. In fact, carbon can make many bonds with many different elements, including hydrogen, as we just mentioned, oxygen, phosphorus, sulfur, and nitrogen. All of those are critically important in producing the organic molecules that make life possible. And all of these different atoms or different elements are going to create compounds with many different chemical properties. Carbon can also create a bond with other carbon atoms, fostering the ability to create small to large chains. Because carbon can create up to 4 bonds, they can create single bonds, not only with other elements like hydrogen, oxygen, nitrogen, sulfur. They can also create single bonds with other carbon atoms. They can create double bonds between adjacent carbon atoms. They can even create triple bonds between adjacent carbon atoms. So the fact that carbon can create 4 bonds and can create bonds with a variety of other elements allows carbon to be the base element for the creation of a lot of different organic molecules. If you remember, organic molecules by definition contain carbon, hydrogen, and oxygen. Polymerization is the biological process of creating polymers. Polymer is a combination of single monomers. So a couple vocab terms, monomer and polymer. Monomers are single subunits, while polymers are multiple subunits. And so what is polymerization? Polymerization is simply taking one monomer and attaching it, usually due to dehydration synthesis, to an additional monomer. When you take one monomer and put it together with an additional monomer, you get a dimer. That is a set of 2 monomers bonded together. When you add a third monomer to the dimer, you get a trimer, which is a 3 monomer sequence. When you add a 4th, it becomes a tetramer, and then when you add a 5th and on, it becomes a true polymer. Why is polymerization important? Polymerization is the step of taking a single building block and creating large organic compounds from those repeated subunits. Carbohydrates are no different. Carbohydrates are composed of carbon, hydrogen, and oxygen atoms, usually in a 1 to 2 to 1 ratio. So when you see carbohydrates written like glucose, which is kind of one of our most basic carbohydrates, glucose has a chemical equation of C6H12O6. Again, that is a carbon, hydrogen, oxygen in a 1 to 2 to 1 ratio. If you were to simplify this, it would be simplified down to C1H2O1. Carbohydrates can be in the form or written in the form of a chain, where you have all of the carbons listed in a chain. Carbohydrates can also be listed as a ring. This would be an example of a carbohydrate chain. This would be an example of a carbohydrate ring. Both of these, this chain and this ring, have the same chemical composition of C6H12O6. C1, 2, 3, 4, 5, 6. H12, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And O6, 1, 2, 3, 4, 5, and 6. If you were to count up all of the carbon, hydrogen, oxygens in this particular ring, you would again get C6H12O6. Organisms use these carbohydrates to store and release energy as well as for structural support and protection. It is really important for you to note that the purpose of carbohydrates is short-term energy storage. Organisms, both prokaryotes and eukaryotes, both plants and animals, use carbohydrates like glucose to store their energy short term. Monosaccharides, these are the monomers of carbohydrates. There are 3 monosaccharides you need to be familiar with. The monosaccharides are the simplest of sugars: glucose, fructose, and galactose. Glucose is the main or most basic simple sugar that all organisms and all cells use. Fructose is a component of sucrose, and again, we're going to get into the disaccharides in a minute, but fructose is one of 2 components of sucrose. Sucrose is the main form of sugar found in plants and fruits. Sucrose happens to also be table sugar. Galactose is a component of milk sugar, which is lactose. Again, is a disaccharide. We'll talk about it in a minute, but galactose is one of 2 components that make up the milk sugar for mammals. Disaccharides, prefix di, means 2, so these would be the dimers of carbohydrates. There are 3 that you need to be familiar with. You have sucrose, lactose, and maltose, and all of these disaccharides are created by dehydration synthesis, which is the removal of water to produce this dimer. So without getting into specifics, you're going to have 2 adjacent rings. You're going to pull one hydrogen off of one of our glucose molecules, and you're going to pull an OH off of an adjacent glucose or fructose or galactose, and those are going to form an H2O molecule, which is water, and by removing one component from each ring, it's actually going to allow each of the rings to bond together. That's dehydration synthesis, and that is the way that all of these disaccharides are produced. Sucrose is table sugar. It is plant-based. We get all of our table sugar typically from sugar cane. Lactose is milk sugar. It is produced by mammals in the mammary glands to nourish their young. Maltose is also a plant-based disaccharide, but is typically reserved for the seeds and fruits portion of the plant, where a lot of germination and fruit ripening needs to take place. Polysaccharides are also produced via dehydration synthesis, although now we have more than 2 of those monomers linked together. Every single one of these bonds would have been formed by a dehydration synthesis reaction, which means every single one of these bonds would have had a water removed in order to link the next monosaccharide to the growing chain. Starch is glucose molecules all oriented in the same direction. There is no branching. All of them have the same conformation, and they are all glucose. Starch would be the functional polysaccharide found in plants. What do I mean by functional? Functional polysaccharides are always the ones that allow for that short-term energy storage, because we said that's the function of a carbohydrate. Cellulose, on the other hand, is all made of glucose molecules, but they are oriented in opposite directions. There still is no branching, but you'll notice the CH2OH group is oriented in opposite directions, which is different than the conformation they had in the starch. Cellulose is the structural polysaccharide, also found in plants, but the fact that I say structural means it is not used for short-term energy storage. It is used for cell wall composition. So plants are going to use cellulose to structure their plant cell walls, and so that's why this particular polysaccharide has a structural use, not a functional use. And then we get into glycogen. Glycogen is all glucose molecules that are oriented in the same direction. However, there is going to be branching that is made possible. So you will see that all of these CH2OH side chains are all going to be oriented up in this particular conformation. However, there are branches that come off, and glycogen is going to be the functional polysaccharide found in animals. Again, functional because it is going to be short-term energy storage. Okay? This is how we, as an animal, store our energy short-term in the form of glycogen. Plants have two polysaccharides, one functional and one structural. Starch, cellulose, both plant-based, glycogen, animal-based. That's it for this particular lecture. If you have questions, bring those to class. Like the video and subscribe. See ya.